Phosphorus is considered a nutrient, meaning that it assists with plant growth. But when it's present in excess in aquatic systems, it can lead to algal blooms. For Stream Team, we measure total phosphorus, which is a measurement of all phosphorus, both dissolved and suspended, within the sample. Elevated levels of phosphorus in stream water can be caused by fertilizer, animal waste, sewage, soil erosion, or atmospheric deposition. Shown here is the equipment that we use for measuring total phosphorus for Stream Team. It's one of our most complicated methods and has quite a bit of equipment. We use 2 pipettes, trays, 2 timers, alcohol wipes. That green box contains vials and a liquid reagent, and then to the left of that is the spectrophotometer that we use for measuring the vials. A piece of equipment that's unique to total phosphorus is the heating oven on the far left, which will heat vials up to 100 degrees Celsius. The method that we use for total phosphorus is actually quite similar to the method that we use for nitrate. As with nitrate, the first step is to open your sample bottle and pipette a set amount into the vial. I've sped up the footage here because the process is relatively straightforward and it's shown in other videos as well. Simply using the pipette as instructed to move a set amount of volume from the sample into the vial. The vials that we use for phosphorus have aluminum foil on the top. Once we've added the sample, we'll peel that off and then flip the cap and screw it in with the aluminum foil side down. As you can see here, there's a powder reagent inside of the cap. Once it's screwed on tightly, you can invert 2 to 3 times to make sure it's thoroughly mixed. Once you've done both steps with all of your samples, you can place them inside of the heating block. Once they're inside, you close it and hit start, and it will heat them to 100 degrees Celsius for 60 minutes. After that step, you can take them out and place them in a nearby fridge. They'll need to stay in the fridge for about 15 minutes in order to make sure that they're completely cooled. Once that 15 minutes is over, you can go and pick them up, and then we can start the next step of our process. With the samples cooled, the next part of the process will look very similar to the initial part. You'll be pipetting a liquid into the vial, except this time, instead of the sample, it's a liquid reagent. It's stored in these bottles with the blue caps, and this reagent is light sensitive. This means that when you're not directly pipetting out of the bottle, you'll want to make sure that the lid is firmly on it to prevent any light from shining in and affecting the reagent. Once you're done pipetting, you'll add a second cap, stored separately, which has an additional powder reagent. You'll screw it on firmly, and then as with the first step, you'll invert it 2 to 3 times to make sure that it is thoroughly mixed. Allow 10 minutes for the reaction to take place. During this time, we'll use one of the alcohol swabs to clean the vials, as we do with the nitrate method. Once the time is up, you're ready to read. Invert the vial once and then insert into the spec. The machine will automatically spin the vial and begin to read the barcode to identify it as a phosphorus vial. It will also shine light through the sample, so we'll cover it to make sure that no excess light can get in. After it takes a moment to read the sample, it will then provide you with a value for total phosphorus. Be sure to record the value, and also note whether the value is negative or under range. Once you've recorded that value, you're done with the sample, and you're ready to move on to the next one.